today, for our expert interview, we'll be speaking with the sort of father of rat laughter research, Dr. Jaak Panksepp. He will share his thoughts on rodent emotions. So he holds an endowed chair at Washington State University's College of Veterinary Medicine and is an emeritus professor of psychology at Bowling State University. He received his PhD from the University of Massachusetts in behavioral neuroscience. And importantly, he is a world-famous expert in affective neuroscience. He employs a detailed understanding of basic emotional systems at the neural level, often using animal and human models in his research. He specializes in understanding how fear, social bonding, and separation processes are organized in the brain. He has published a remarkable 300-plus scientific articles, co-edited a multi-volume handbook of the hypothalamus, as well as for emotions and psychopathology. And he's the author of a text entitled Affective Neuroscience, The Foundations of Human and Animal Emotions. His work has received well-recognized merits, including, for example, among many, the NIMH Research Scientist Development Award. So we have a special pleasure today to speak with Dr. Panksepp on the emotional life of rodents, animal models of human brains. So welcome, Jaak. Thank you for speaking with us today.
Thank you, June.
What I wanted to start out with was asking you a bit about sort of where your journey in the field began. In other words, what first got you interested in studying emotion?
Well, I think we all have emotional lives, so any intelligent person is very interested in these mysteries. They've been a traditional topic of conversation ever since we could talk. Of course, talking is not an evolved function. We learn how to talk. So we have many different words for it, but it became very clear to me when I first entered graduate school in clinical psychology that there was not a conversation about the real nature of emotions. And I realized at that point, at the very beginning of the cognitive or neuroscience revolution, that one really had to understand these at the neuroscientific level. And really, the precipitant for me was working in the back wards of a psychiatric hospital when I was an undergraduate and seeing the people coming into the hospital in emotional turmoil and becoming fascinated by this human drama. So I wanted to be part of it. And that late in undergraduate work, the best route was clinical psychology, but that was the day of behavioral modification, and talking about emotions was not permitted.
So since then, I mean, you've just pioneered a whole field of research. I mean, you've played this role in not only founding, but coining this term affective neuroscience, that we use so widely today, devoted to understanding these emotional processes through psychophysiological tools. And being a founder of this field, coming from a time where emotion was not a sort of, allowable phrase, I wonder, as you see the field, when it's emerged and sort of where it's come now, what are some of the most fundamental discoveries we've made through affective neuroscience?
Well, I think the most fundamental general principle is an evolutionary one. We will never understand emotions in human brains and minds unless we take an evolutionary approach. And we have to envision the brain as an evolved organ with many layers of development. And if we don't bring these layers of development into the conversation, we will be talking past each other, which we've been doing ever since I entered the field. Psychology has become very excited about emotions, but they're working at what I call the tertiary process level, namely emotions that have already been processed by complex cognitive processing, which we're very skilled at. That's a cortical function, but practically everything in the cortex is learned. Below that, you have primary processes. Behaviorists call them unconditioned responses, things activated by unconditioned stimuli. They did not study those, but there are a host of emotional systems down in the brain that we share with the other animals. If you take an evolutionary approach, you cannot avoid that conclusion, and affective neuroscience started with that. So you've got tertiary process on top that you cannot study in animals. You've got primary process at the bottom, which you can't study very well in human beings. And in between the two, you have what I call the secondary process, which is learning and memory. And all behaviorism was based upon having solid tools for learning and memory. That's very important, but that's where the unconscious resides. The unconscious does not reside at the bottom of the brain. There you have affective processes, and that is a very difficult lesson to penetrate through the jungle of words.
So given, you're talking, and it's very important, this jungle of words, as you say, I mean, that may have gotten in the way of some of the most important insights that need to be brought out in this field. From this, sort of what breakthroughs would you like to see in this field?
Well, first of all, I think I would like to see everyone acknowledge that if we do not take levels of control into account, we might be talking past each other. And I think everyone has to accept the data that comes from the animal research, especially when it converges on very robust predictions for our own species that we're all most interested in. I certainly am. I entered in clinical psychology, for heaven's sake. And I realized that without a foundational science, things will not move forward, including new medicinal development, which we are very interested in. So from my perspective, one can ask, what do we find at the foundation of the emotional mind? And we know we find at least 7 types of not basic, I would call them primary process emotion, a slightly different concept. Namely, we can produce coherent emotional behaviors by stimulating specific parts of the brain in every mammal that has been studied. This emotional display is instinctual. It has autonomic components, it has clear behavioral components, and we're only putting garbage into the brain. Just electrical stimulation out of the wall socket brought down to very tiny microamp levels, and you get coherence out. That automatically means the coherence was built into the brain. And the great breakthrough of affective neuroscience is to ask, does the animal like it or dislike it? Is it good or bad? I mean, the behaviors view would say the animal doesn't care. It's just a puppet. These are just actions. There's no psychological content. But you can ask the animal, when I arouse this emotional pattern in you, do you like it? Namely, is it a reward? Do you dislike it? Is it a punishment? We've got the tools for that. And there's a law of nature that for some reason we are still neglecting in psychology. Wherever in the brain you activate an emotional process, it's always rewarding or punishing, period.
I mean, I just think that's so true, and I think many of these insights are ones that we need to really take with us as we move forward in this field of affective neuroscience, which is now just, expanding, even exploding at fast, proportions, with lots of new tools and discoveries. I wanted to turn to another really important part of your work, which is where you've scientifically demonstrated something that a lot of people maybe believed wasn't possible, which is the fact that rats can indeed exhibit laughter, this kind of special chirp. And I wondered if you could tell us a bit more about the supportive evidence that your really, really insightful work has done that suggests that rats can laugh.
Well, we were looking for other clear primary process emotions, and it struck us that play might be one of those. And we developed very rigorous procedures for the first time to study play in the laboratory. And it was wonderful. The animals loved it. They became very resilient to depression. It was as powerful a reward as food for a hungry animal. And it looks like our own children playing. And at one point, we started asking, are there play vocalizations? Now, we know that rats communicate in the ultrasound range, so predators will not be able to detect them. And lo and behold, with ultrasonic equipment, the playing field was full of little chirpy sounds. And we called them play vocalizations. And we studied them, especially Brian Knudsen, who's now at Stanford and part of this project. The playing field is full of these little sounds. And lo and behold, one morning I woke up and I said, what if that is laughter? And it took years to come to this insight. And I think it came from a dream. So I immediately went to the laboratory. Jeff Bergdorf was waiting for me. I said, Jeff, let's go tickle some rats. He said, okay. First animal chirped like crazy when we tickled them. The second animal, third animal. We could bring this vocalization under stimulus control, and now it's our primary measure of positive social affect because Jeff, in his dissertation, mapped the system with deep brain stimulation, and every place we were able to get a chirp, the animal self-stimulated that site. So we have the first validated measure of social joy that's based on neural circuits as opposed to just theoretical speculation.
So in what way do you see rat laughter as similar or distinct from human laughter?
Well, it's homologous. There's an evolutionary continuity. Nothing in evolution is identical. A lot of people accuse me, how can it be identical? That's nonsense talk. Homology means there's an evolutionary relationship, and there are bound to be similarities in the mechanisms. For instance, the human brain contains no neurochemistries that are not present in the other mammals. So we are not unique at that level. We're only unique because we have an enormous neocortex, and that neocortex is empty at birth. Everything is constructed there, including vision. Vision has to be learned in the neocortex. So I'm a constructivist in the neocortex, but at the bottom of the brain, we have to understand the instincts. And the instinct of laughter in humans appears to come from those same circuits. People that have, gelastic laughter, that is caused by epileptic fits. That is triggered down deep in the brain, very much in the areas of the brain that we map for rat laughter. So we think by working out the chemistries of rat laughter, we will have new ideas for antidepressants.
Yeah, I wanted to ask you, what do you think this can tell us about what ways we can understand social joy in molecular terms and develop, as you're saying, novel, treatments like antidepressants?
Well, what we did with my colleagues at Northwestern University, Joe Moscow's group, we did microarrays on the neocortex, which is not needed for play, but which play influences. Play programs the neocortex for being a higher social brain. And we found that the microarray analysis yielded 1/3 of the genes that we measured being modified either up or down. And we had a very simple strategy. Whatever gene is upregulated, and the top one was insulin-like growth factor 1, might be a molecule that promotes social joy, and insulin-like growth factor does. The second one, but it also increases cancers, so it's not a good target for medicinal development. But the second one was the NR2B subunit of the glutamate and glycine receptor complex, and that is a target that doesn't have any medicines. And lo and behold, we developed some molecules to tickle that site, and it elevates 50 kilohertz chirps, which is what we want. We want an antidepressant to elevate positive social feelings. So we pursued that molecule first preclinically, namely animal research, and it's now gone through human testing. They went through phase 1 toxicology. It was safe. Phase 2A, which was the first feasibility study, and it produced robust antidepressant effects, namely bigger effect sizes than the SSRIs, which have enormous problems, SSRIs. If you're on them for years, you get addicted to them, and you can't get off them, which makes it a wonderful money mill. So we have a molecule in development, and the whole philosophy of affective neuroscience is when you see the similarities of the animal, the other mammals, and the human brain, you can work out the details that you can never do in humans.
Wow. So what do you think we can also learn in terms of molecular terminology about things like sorrow? We're talking about joy, and there's always sorrow as well as part of our emotional life.
That's a wonderful question. I'm glad you asked it, because the very first set of major studies that we did when we open up the Pandora's box of affective cross-species neuroscience was to try to identify the brain systems that mediate social attachments, the kinds of systems that Harry Harlow had shown how the motherless monkey is a very sad creature, but it cannot function in the world. And we thought there might be a separation distress system in the brain, and we said we can monitor this by looking at separation calls. You separate little one from the mother, and lo and behold, we found the system. It's a very robust one in many mammals, not laboratory rats, by the way. And that's a long story. A lot of people are making mistakes. It's been bred out of the laboratory rat. And we found first that it was modulated by endogenous opioids. And we had this theory that social attachment is an addictive process, that many people who have psychological pain because they're disenfranchised, they don't have good social networks, they're continually feeling bad, and they start to self-medicate themselves with opioids because the rest of us that have happy families are releasing opioids all the time during positive interactions like play and, just hugging, love, sex, lust, all the social positive things facilitate opioid activity. So, we've demonstrated that this separation system actually has opioids around it, and touch activates it even. So, a little baby that's being cuddled, you're releasing their opioids and you're producing a social addiction. Addiction is a bad word there. This is love. There's probably more evidence that opioids mediate love more than oxytocin. Oxytocin also is very effective, but oxytocin increases confidence, which every mother needs. If you have this baby coming and you don't have confidence, you're not going to be a good mother.
So this is all really interesting. I mean, thinking about the field of affective neuroscience that you played such a fundamental role in founding, transitioning to understanding social joy and the molecular terms of it and studying it in rodents. And then now thinking about sorrow and how we can use some of these insights to help develop novel kind of targeted treatments. When you think about sort of where the field started, where it's come, how do you project into the future in terms of thinking, well, what's in store for the future of emotion here?
Well, I think the really wonderful aspect is that we finally can scientifically understand the very foundation of the human mind by studying other animals. The animal research will not reveal how our upper mind works, but if we take an evolutionary point of view, we have to realize that what came first always rules. These solutions for living that the primary processed emotions are constituted from, these are foundational for learning and memory. So we had a law of effect, Thorndike. We should now have a law of affect, exactly the way Thorndike originally phrased it. It's the affective circuits of the brain that control learning. And we have to realize that these fundamental affects are tools for living. And the way nature solved projecting itself into the future is to generate these spooky things called feelings, affects, that tell you immediately whether you're on the path of survival or destruction. So every good feeling indicates you're on the path of survival, just statistically. And all the negative feelings tell you you might be on the path of destruction. This is anticipating the future. That is the function of affect. And then you anticipate through learning and memory. So affect controls learning and memory. And learning and memory give you the tools for thinking. You have to have facts before you can have ideas. But the ideas are the creation of our higher mind, and they can be anything we can imagine, and that's why there's so much controversy at the verbal level. So if we accept the foundations, we can have a clear discourse, I believe.
So what advice do you give then to future students who are just thinking about embarking in the study of emotion or just getting into their first few years?
Well, I think I would suggest that they immediately read Affective Neuroscience 1998. I spent 10 dozen years writing that book, and I wrote it for other psychologists so they would be attracted to the neuroscientific analysis. And I just came out with archaeology in mind to make it a bit simpler, updated, especially for clinicians. And I was delighted. The first community of psychologists who were interested in it, in this vision, were clinicians. So that was very gratifying for me. And I would say that every future generation of psychologists has to realize that you cannot be a real psychologist unless you understand the brain and you're able to navigate in your mind how the brain is organized. That has to be the foundation of psychology. I argued for this starting in the 1970s when we had the Utah Conference on Graduate Education, but it still has not happened. I don't think we can graduate psychology majors who have not taken neuroanatomy and functional neural processes. That is foundational, just like DNA was foundational for biology.
So it seems that what's most important is to truly understand the human brain if we're to understand anything about emotion.
I would say we cannot understand emotions without the brain analysis. That does not neglect or negate higher level analysis. That is very interesting. But remember, every human being is interested in the higher part of it. We would not have art. We would not have theater, music, novels. We would not have any of the riches of, the arts, which are so important in our lives, without emotions. But the arts are beautiful creations of the basic emotional tools that we have. But the symphony finally is created by the full brain. And of course, we have the most rich and complex upper emotional life of any creature.
Well, on that beautiful note, I want to thank you for speaking today. It was a pleasure to talk with you.
Wonderful to talk to you. Thank you, June.
Thank you. So this concludes our Experts in Emotion interview series with Dr. Jaak Panksepp from Washington State University's College of Veterinary Medicine and Emeritus Professor of Psychology at Bowling State University.